The following program is brought to you by Caltech.
Welcome. I'm Peter Groom, class of 75. Welcome to the 76th Annual Caltech Seminar and Reunion Day. The first speaker today, I have the pleasure of introducing Viviana Gradinaru, who is actually went to Caltech here for undergrad. She then went on to Stanford University and got her PhD there, and is now a professor of biology here at Caltech, and she's going to be talking to us about brain control with light. So please give a warm welcome to Viviana Gradinaru.
Hi. Good morning. Thank you for the very nice introduction. So glad to see all of you here, and also a little tense because it's nothing more difficult than to talk to Caltech alumni. As I learned during one bioengineering lecture, they tend to be very quiet and observant, so please interrupt. That would make my job much easier. So as a Caltech undergrad and afterwards, I was many times asked to summarize my Techer experience in one or two sentences. And this was always very difficult for me. How do you do that? Because it's the academics, the life, the intensity, so it was very difficult. Until last year when I came here for a short visit with my construction team for the lab, and afterwards I took my son to see the houses. And we were in the grassy area between Fleming and Ricketts, and we were just looking through the houses, and then two gentlemen came, and they looked at my son, and with an affirmative nod, they say, they admit them earlier and earlier, right? So that was fun, but it also got me thinking. That was it. What in my experience was special about Caltech was the trust that was put in you regardless of the variables associated with you. That put a lot of freedom. It gave you the freedom to focus on learning and creating. It also gave you the resources. So not the academics, not the intensity, it was the trust that was put in you and the lack of judgment. You could have many variables, you could be at very different spectra on all sorts of features, but you are still allowed to focus on learning and creating. So that's what made it special for me. So in our lab, we like to learn about the brain. And we've learned some things, as I'll tell you about today, but this is just scratching the surface. And part of it is because of the lack of tools. So in our attempt to understand the brain, we are guided by Feynman's words, who famously said that if you want to understand the marvelous biological system, you should just look at the thing. In his mind, probably was all very simple. Just look at the thing. But this short sentence has many layers. To look at the thing, you need the tools necessary to allow you to look at the different scales and the different dimensions. You need to know what to look at, and once you've acquired all of the data, to make sense of the data. So what we do in our lab is to make some tools to understand the brain, and then generate the data and try to understand how some of those data summarize the behavior and what we are. So without further ado, I'll go into our research. The brain has many different cell types, and this makes us complex and beautiful, I would say, but it also makes it difficult for neuroscientists to understand what's going on. So if you look even in a very small brain structure, like the hypothalamus here, very tiny brain structure. This has been involved in all of these different behaviors that you see here. And if you look even further at the cellular level, there's very different cell types. So now we have the ability to stain these different cell types. So what you see in green here are cells that were shown to be involved in social bonding, and what you see in red are cells that were shown to be involved in thirst. And what you don't see in black is yet other cell types that are involved in all of these different behaviors. To understand their contribution to behavior, neuroscientists have access to a few tools. They can do chemical ablation of certain populations and understand if you remove a certain neuronal type, what behavior do you lose or what behavior do you gain, and you would rather not gain. Another alternative is to apply an electrical field. Neurons are susceptible to electrical field, and then they would respond, some of them in inhibitory ways, some of them in excitatory ways. However, for both of these methods, chemical and electrical, the big drawback is that there's no specificity, or the specificity is very difficult to achieve. So what you get here is a combination of the neuronal responses, making it difficult to understand. So why is it important for us to know what cells should be modified in order to affect the behavior? It's important because in neurological disorders, and here is pictures of brain of a person that has Parkinson, you can interfere with electrodes. Here is an MRI of depth electrodes implanted in a motor area of the brain. And these electrodes are used to deliver 100 hertz frequency to the brain. And miraculously, this actually eliminates tremor and allows the person to walk, literally walk the next day after the surgery from the hospital and home and live a better life. However, because of the non-specificity of the electrical stimulation that I mentioned, there's also side effects that you would rather avoid. So some of the work with translational relevance in our lab is focused on just this, to try to understand how electrical stimulation in human disorders work and to try to create the tools for this. So what kind of tools could one design in order to tackle such problems? We are focusing on using light, on using photons, because light can be very versatile. It not only has the advantage of growing the biological systems, but in biology, we've used light to observe biological systems, to observe organs, cells, proteins, DNA, RNA, and further, at the very, very fine levels in very different cell types and circuits. And newer work actually uses light to control cells, and this will be the bulk of my talk. How can we use light to control cells in a specific fashion? And towards the end, if there's time, I'll show you a very new application where you can use light to image very large brain areas, despite of the optical scattering of the tissue. So how can we render electrodes neurons responsive to light? In the brain, there's not easy access to light, as you can imagine, so there's no evolutionary reason for the neurons to develop such tools. This turns out to be an advantage because we can introduce proteins into neurons that are light responsive. And the way to introduce these proteins is by genetic targeting, where we can decide, okay, this light responsive tool would go into this cell type and not the other cell types. And we can do this by modifying the DNA sequence in front of the light responsive tools. So what I'm showing here is a comparison between the electrical stimulation case, where the field will affect all sorts of neurons in the vicinity of the electrode, versus an optical control, where you have to apply the light to the entire tissue, but only the neurons that were sensitized, that were modified to respond to light, will actually be controlled. And the way we do this is with a channel. Here, it's called Chlamydomonas reinhardti, and it's a cation permeable channel. And when it absorbs blue light, photons, opens a pore and allows a flux of sodium. Naturally, neurons, by a flux of sodium, get excited. And a nice feature of this is that it has very, very quick kinetics, millisecond scales. So you can pulse the neurons at 10 hertz, 50 hertz, 100 hertz in a very cell type specific fashion. And then you can activate circuits in the brain and see how this impacts the behavior. Excitation by itself is not enough. Many times you would rather remove a circuit and see what happens when you lose that circuit, as it happens in many times in the diseased brain. So for this purpose, what we have is a chloride pump here that's responsive to yellow photons. So when yellow photons are absorbed, chloride gets pumped into the neuron, and the neuron turns silent. And by using the same trafficking methods, you can introduce the chloride pump only in the cell types of interest. I briefly gave an overview of the technique. Now, before I go into the details of how we created this technique and what challenges we had, because it was not straightforward to find these tools and make them work well in mammalian systems, I want to give you a feeling of what you can do with this technique. In my laboratory, we use rodents as animal models because we feel is a good proxy for the human brain, and we can still perform the manipulations to understand. non-invasive experiments in humans can only give you this much information, and the smaller animal systems like the invertebrates are very good for genetics. But we chose a compromise between the human brain, which is the ultimate goal, and the simpler systems, so we chose to focus on the rodent model. So what I will show you next is a movie of a mouse that was modified to express these light-responsive proteins in a region of the brain that in humans by fMRI studies was shown to be activated in fear response. So this region is the amygdala. And we introduced channelrhodopsin, the excitatory protein, into the amygdala, into the fibers, in a very specific circuit. So we didn't introduce in an indiscriminate fashion throughout the amygdala. We chose excitatory cells in a subnucleus of the amygdala. And then we stimulated those cells at 20 hertz, and we saw the effect on the behavior of the rodent. In the rodent world, there's this behavior called the elevated plasmase that's commonly used to measure anxious behavior or stressed behavior. And the way the test is, you have, as you can see here, a plus sign with two, maybe I should pause this. So you have two open arms. There's no walls. So you have a plus sign. Two of the sides have no walls, but two sides have walls. Rodents are naturally anxious creatures, so they prefer to stay in the areas with the walls. And they wouldn't venture on the outside because in the natural world, this is associated with having predators around you. So without any modification of the neuronal circuits, the rodents would prefer to stay in the enclosed sides of the maze. So this is the control period. No modification of the circuits. And you'll see that the rodent pretty much prefers the confinement of the closed arms. The optical stimulation will be indicated by blue writing on the side. And you can see what change is being induced by the modification of the circuits. So what you've noticed is that now the rodent feels comfortable enough to venture into the open spaces, which is something that without the modification of those neurons in the amygdala wouldn't naturally do. Now the light is off, and what I find interesting in this case is that the rodent, as you'll see, now is restricted to the closed walls, to the closed spaces, despite having the information that the open spaces are safe. So this is something that is very interesting to me. The rodent was just there, knows that that's a safe space, but still the internal drive to hide is so strong that wouldn't go there. So this is what we can do with the tools that we developed in our lab. Now I want to take many steps backwards and tell you about how we designed the technology and what challenges we had to make it work to this level, because this is where the fun was. So their origins are very interesting. They come from, so they're called, the class is called microbial opsins, and they come from unicellular algae or bacteria. And for example, here, the experiment that I showed you used the protein from this green alga, Chlamydomonas reinhardti. And then the inhibitory tool came from a bacterium in a very salty lake in the Sahara Desert. This is a population, it's called the social alga, and there's another tool that came from there, also an excitatory tool. So what we aimed to do was to extract these proteins or the genetic code for them and use that genetic code into mammalian systems and also into invertebrates, so in animal models such as worms, C. elegans here, very, very good for genetics, and flies. And then move them to rodents, and there's even primate experiments that can use optogenetics. However, by doing that, we ask a lot from the protein. We ask that the protein that functions well in its natural environment would keep its function in a very different system. And it's been difficult to do that for some of the proteins. I'll start with the success story. The excitatory opsin, miraculously enough, we could just use the natural sequence, very few modifications, and we were able to generate transgenic animals. So these animals are being born with a light-sensitive protein in their brain of microbial opsin origin. And they're just fine. So they live a normal lifespan, they breed normally, and they have normal behavior, which is rather remarkable. If you look here, the opsin is tagged by YFP, and you can see its distribution throughout the brain. Another bonus of this technology, in addition to the fact that you can very precisely stimulate or inhibit the circuits of choice, is that you can also get real-time feedback of what you're doing to the brain. It's not a black box. Many times the behavioral experiments, you deliver a drug to the animal, you measure behavior, but you don't know what changes are in the circuit. And same with electrical stimulation. It's very difficult to get real-time feedback because of this simple technical challenge. Each electrical pulse will create a stimulation artifact. So if you try to control the system at very high frequency, you won't be able to tell your signal from your noise. Fortunately, in the case of optogenetics, because the stimulation is optical, this artifact is much, much smaller, and you can tell the signal. So here is a simple experiment to illustrate this point. We took the transgenic animals that I told you about in the previous slide, and then we made this very simple device, a fiber optic to deliver the light, and then an electrode to record what we are doing to the circuits by controlling this opsins. And this is the signal from cortical layers. Each pulse of light, notice the time scale here, millisecond time scale, elicits population activity with very good temporal precision. So you can control the circuits, and then you can understand in which ways those circuits are responding, both by the electrophysiology and behavior. So now the challenging part, the inhibitory tool. As I mentioned, it comes from very different species, from bacteria. And what we've noticed because of this evolutionary distance, when we express the protein into mammalian neurons, we would get aggregates. And these aggregates kept growing and poisoned the cell. So we couldn't really use the tool in its natural form because it would have these aggregates that interfere with neuronal function. So there was a lot of motivation into trying to bypass this challenge. And first, we needed to know why this is happening. And we performed stains for intracellular organelles. And what we noticed was that there was 100% overlap of the aggregates from the protein with the endoplasmic reticulum. And this was, in a way, the aha moment, because the bacterium doesn't have the endoplasmic reticulum, but the mammalian system has. The endoplasmic reticulum is the protein factory of the cell, so all the proteins have to go through there. And in mammalian cells, they have precise instructions where to go and how to go, and they have signal peptides that tell them where to go. And this microbial opsin protein didn't have those signals. So the strategy became relatively straightforward. Let's look at trafficking signals from mammalian proteins and take those trafficking signals and transplant those onto the protein, onto the microbial opsin, in a hope that now the mammalian cells would recognize the foreign protein as their own and process it appropriately. So this is what we did. And this is the result. So through a few iterations, of course, I'm not showing you a lot of the failed experiments here, but what we did was to attach a sequence for endoplasmic reticulum export, and we abolished the aggregates, and then further we attached a sequence for membrane insertion. I failed to mention that these opsins, to be functional, they have to be in the membrane of the cell. So the end result here was an opsin that was modified to express very well on the membrane of the cell. And the result was that the currents, the photocurrents associated with the stimulation would be much more potent. So what you can see here is a neuron inhibited over 15 minutes, and this was performed by electrophysiological recordings. These opsins don't respond to a discrete wavelength, they respond to a Gaussian. And interestingly enough, because we increased the membrane expression and the efficacy of the opsin, now we could use red light to inhibit the cells. And this is important for two reasons in our experiments and potentially for further translational applications as well. Red photons are much safer. They don't damage the tissue as much. And also, they penetrate more. So if you want to control larger brain areas, you'd rather use red photons rather than blue photons. And the other advantage is that the excitatory tool that I talked about responds to blue light. So now having the capacity to control the inhibitory tool with red light, you can actually use both of them in the same cells. And you can either use blue light to excite the cell and use red light to inhibit the cell with minimal crosstalk. So that's an added bonus. And this also illustrates the same point, the need to have many different opsins that respond to different wavelengths. And because their spectra is very broad, it's been challenging for us to achieve good separation. And in the brain, that's a an important need because there's many different types of circuits that could benefit from being controlled. So some of the work, of the ongoing work in my lab is trying to mutate the existing opsins and mine the natural diversity for more opsins that would respond to different wavelengths, have sharper activation spectra, or have different ion permeability, permeability for calcium or for other biochemical factors. So how are we on time? I think we're good. So I want to now spend some time on the translational applications or the clinical applications of this technology. And I will tell you about one story where we used optogenetics in the circuits in a rodent model of Parkinson's disease. And what this experiment told us is that there were alternative pathways involved that otherwise would have not been identified. So as many of you know, Parkinson's disorder is caused by the death of dopaminergic neurons in the substantia nigra pars compacta. And this dopaminergic loss causes a lot of circuit malfunction, so abnormal activity in the connecting nuclei. And if we look here at an oversimplified diagram, dopamine in the substantia nigra pars compacta has a beneficial effect for movement through both the direct pathway in the striatum and the indirect pathway. Now, when we lose dopamine, such as the case of Parkinson's disorder, a lot of the downstream nuclei show perturbed activity, such as high activity in the subthalamic nucleus here. And the end result is a decreased motor output. So now some alternatives have been tried, such as supplementing the dopamine, which works quite well in the short run. However, after 5 years, the need to increase the dose and the dyskinesia associated with it prevents further use. An alternative that works very well is deep brain stimulation, as I'll show you a video. And what deep brain stimulation does is to apply an electrode here into the subthalamic nucleus. In the disease state, subthalamic nucleus is hyperactive. The electrode excites, so the stimulation is 100 hertz, this nucleus, and then the patient feels much better. However, the mechanism is not known how this works, and it's rather controversial whether the stimulation itself shuts off the nucleus or further enhances the activity, so that's not known. And it's rather important that we understand the mechanism because of the tendency in the field nowadays to use deep brain stimulation for other conditions as well. So in '93, it's been introduced for Parkinson, and it's been FDA approved for other motor disorders since then. Many patients benefited from it. And there's more sophisticated iterations now. So there's a patient-controlled DBS, so the patient can control its own stimulator. However, there are side effects, and this same method is being is shown promise for OCD and depression as well. So let me first show you, if this will play okay, fingers crossed, a patient that can control its own stimulator. See the magnet here implanted in the under, so that's the reason why the gentleman is shirtless. So now I turn the stimulator off. So now it takes the controller and turns on the deep brain stimulator, and it can move and walk. So a few things you notice, the gentleman is rather young, and also the quickness of the onset and offset of the stimulator effect. And he can do this by just touching the battery pack here in the chest. So it works so well. But there are complications with hemorrhage with the implant. But for all of these patients listed here, and it's been more, this is the statistics that I could find published, but it's been more since then. And there are many thousands of articles about this topic, how does it work? And as I mentioned, it's been FDA approved for obsessive compulsive disorder, and it's considered for depression. So the big question is, how does it work? And we try to understand this by using optogenetics, because electrical field will affect everything in the STN area. So we don't know if it's inhibitory cells, excitatory cells, support cells such as the glia, or even other nuclei in the region. So our approach was going to be very systematic. Let's put the inhibitory and excitatory opsins in different cell types and in different nuclei in the stimulation area in an animal model of Parkinson and see which circuit has to be modified and in each way to recapitulate the effects of the brain stimulation. So we started with a straightforward experiment. I mentioned that in the Parkinsonian brain, the subthalamic nucleus is hyperactive because of the loss of the dopaminergic neurons. So the obvious experiment would have been to insert the inhibitory opsin into this nucleus and try to shut off the nucleus in an attempt to restore the behavior. So this is what we did. The way we can do this is by using viruses. They're very good at infecting cells in the laboratory setting, and we modify their capsids so they're not replicative and not infectious for us. So we can package the opsin DNA into a virus and deliver it to the subthalamic nucleus here. And if we focus on the area, some of the cells will will express the opsin DNA, and then the ones that have the opsin and the light will be inhibited by light. So here is how the brain slice from this experiment looks like. In a bright field, you cannot tell which one is the subthalamic nucleus and what's the surrounding area because the brain is heterogeneous and has no marker. However, you can see here remarkably, we modify the opsin DNA to have a specific promoter, which is a tag for excitatory cells. And this nucleus here is a nice feature of being a mainly excitatory nucleus embedded in an inhibitory network. So you can genetically extract its shape here and see the expression of the opsin only in the target nucleus. So now when we apply the fiber optic, we are confident that we are controlling only these cells in the system and not everybody else, as it would be the case when you put an electrode here. We confirmed that the opsin can inhibit the otherwise hyperexcitatory nucleus, so you can record with the system that I mentioned before, and this is the neuronal activity before light inhibition and during the light inhibition. And then we perform the behavior. The test is a unilateral rotation test, and what's important here is to note that there's no difference. So there's a Parkinsonian rodent model, but the modification of the cells in the subthalamic nucleus didn't cause any change in the behavior. However, the electrical stimulation returned the animals to their normal baseline state, which was no Parkinson's symptoms. So this was very bothering because it was the experiment that was the most obvious to try, but it showed no difference. So the result was that reducing the activity in the subthalamic nucleus would not mimic the effects of DBS, which told us that other players might be involved and made us look for them. So after many failed experiments, all that focused on the nuclei very close to the target. So this is the DBS target. And we've attempted to control all the cell types in the STN and the cells in the neighboring areas. All of those results showed no improvement. And then we looked more carefully at the diagram, and we realized that there is very strong projection from cortex to the subthalamic nucleus that's not being modulated in our experiments, but that's very likely to be affected by the electrical stimulation because the axons are right there, and the action potential from the neuron can backpropagate and activate entire circuits in the cortex. And this was a circuit that couldn't be studied by conventional methods. You needed a cell type specific technology in order to address this. So we decided to use optogenetics to focus on this pathway, cortex to the subthalamic nucleus. And also, please note, he has a reciprocal connection here. So what we did, we were fortunate enough that the transgenic animal that I mentioned before had a very interesting feature. In the subthalamic nucleus itself, unlike the picture that I showed you before where the cell bodies were expressing the opsin, here you see no opsin in the cell bodies. Rather, you see the opsin in the axonal fibers. And this gave us an experimental model to control the axons in the subthalamic nucleus and leave the cell bodies alone. And the result was very dramatic, both in the electrophysiology. So I should mention, when the neurosurgeon performs the deep brain stimulation in clinics, if the stimulator is at 100 hertz, the tremor goes away, and the motor behavior is normal. But if the stimulator is instead set at 20 hertz, so nothing else is changed, the same area, but if the stimulator is at 20 hertz, there's more tremor. and impaired movement. So it does make a difference what kind of stimulation you're using. When we recorded in our experimental animals, what we saw is that high-frequency stimulation at 120 hertz would reduce activity, and very interestingly, low-frequency stimulation at 20 hertz would have the opposite behavior, which gave us a proxy for the human case as well, where the frequency of stimulation does make a big difference. So then we performed the behavior on these animals. So high score here means impaired behavior, low score means normal motor behavior. And what you can see is that all of the animals reverted to better motor behavior during high frequency stimulation. And then this effect went away once the light got turned off. And interestingly, for low-frequency stimulation, the opposite was true. Please note the numbers here on the scale. This was an overachiever, but everybody else was also significantly different. So, yeah. So now the puzzle was, OK, which axons are we affecting? What if it's the case that there is a cell population that's more superficial that could be modulated and give the same effects? That would be of great benefit, especially for the patient category where hemorrhage is a big concern. And usually it's a big concern the more advanced the state of the disease and the age of the patient. So for this project, I collaborated with Jamie Henderson, a wonderful neurosurgeon at Stanford, and he told me that the inclusion criteria for this technique that works very well is minimum likelihood of hemorrhage. So this excludes a lot of the population that could benefit from the therapy. So after talking about these results, we got very interested about finding this putative superficial population that could be modulated. And we knew that there is, as I showed you in the diagram before, that there is a projection from the motor cortex to the subthalamic nucleus. And we confirmed this projection both in terms of anatomy and in terms of electrophysiology. And what we noticed was that every time you stimulate the STN, you can get activity in the motor cortex. So this suggested the alternative experiment. Rather than stimulating deep within the brain in the subthalamic nucleus, let's attempt to stimulate superficial cells and see if the effect is similar. So in the mouse model of the disorder, much to our surprise, stimulating the motor cortex, in this case, not the entire motor cortex, layer 5 excitatory cells. The animal was modified in such a way that only layer 5 excitatory cells would express the opsin. So in the high frequency case scenario, the motor symptoms were ameliorated. However, note that low frequency didn't make a difference, which tells us that motor cortex stimulation is not 100% identical to STN, which it shouldn't, because they're different locations. But what was interesting was that the motor behavior was affected. And at this point, the always skeptical neurosurgeon said, oh, this is great, but it's a mouse. Do you even have this connectivity in the human brain? Because the rodent brain is a proxy, is a good one, but a lot of things are not the same. So he obtained permission in his patients to do diffusion tensor imaging of the axonal fibers in his patients, and also do recordings to check for this pathway. And this is just a summary. So this is the scan from a Parkinsonian patient that Jamie did. And what this shows is axons from cortex to the subthalamic nucleus. So this image from a human brain confirms what we call hyperdirect pathway from the cortex to the subthalamic nucleus. And he also performed recordings with arrays on the cortex that show that when the electrical stimulator was on in the STN, he would see changes with very short latency in the cortex, showing that this pathway exists and it's functional. So now the intriguing possibility is, can you perform more superficial stimulation? And attempts have been done. The difficulty is that the cortex performs many different functions, and the specificity can be a concern. While in the STN, this can be a focal stimulation point, which would have fewer side effects. So the clinical work is continuing at Stanford, but my lab is very much dedicated to basic science. We are always interested whenever we have interesting or relevant results to communicate with our clinician friends. But in our lab, we wanted to focus on improving the tools for this purpose so they can actually help further. And one tool, as you can appreciate for here, and this will be my final point, a missing part that's very important in neuroscience is connectivity information, especially from very large brains. This pathway was there, but it was not noticed because we didn't know to look for it, first of all, and it's very difficult to reconstruct, especially in the in vitro brain. In our experiments in mammalian brains, we would like, ideally, to see the pathways. And the way we do this currently is to fix the brain and generate very thin slices, as thin as 40 micrometers, and then image each single one of those slices, then reconstitute the images. But what you can see here, these are fine axons. So if you do your slices, reconstitution becomes a computational problem. And also the chance of misalignment is a very large one. So I want to tell you about a technology that was very recently published, and I was involved in it at the beginning at Stanford, that allows you to image through the entire brain without slicing. And this can have very significant implications for neuroscience because you can modify circuits and then image them. This method not only allows you to see the circuit without slicing, but allows you to stain for molecular markers. So allows you to stain for different cell types, for activated proteins, for inhibited as well. So what you see here is, and let's see if the movie plays, it's a chunk of the brain around the hippocampus, which is an area involved in learning and memory, and it's stained. So this different cells here are stained for different markers. You have the excitatory cells in green, then you have their nuclei, and then you have the different markers here in different colors. So the way this method was possible was by making the brain transparent. When you try to image the brain, the light scatters in all directions after a certain depth, so it's very difficult to reconstitute a low-L image. And this is because of the lipid content of the brain. So this method, the first author of the paper, Chang, extracted the lipids out, which by extracting this, you can have a more transparent brain. The problem then became the tissue would collapse because it didn't have the support for the cells. So the trick was to infuse monomers. So before extracting the lipids, infuse monomers into the brain. And these monomers would cross-polymerize temperature-based. So it would offer a support for the cells. And then the lipids were extracted. So now the artificial structure would still hold the circuits in place. And this provided enough transparency to the brain to be able to image the entire brain. So in a way, ironically, now the limitation is the working distance of the objectives, not the transparency of the brain. So I want to show you, that might take a while to play. Okay. So I have to emphasize this is not a sliced brain, it's not a reconstituted brain. The chances of misaligning things is practically canceled. And you can do this for the entire brain, and you can do this for human tissue as well. There's precious human tissue in the brain banks that hasn't been looked at because of the limitations. So, okay. So it's rather exciting for neuroscientists. The combination of optogenetics and the ability to reconstitute the circuits in vivo gives us a lot of work to do, which we're always happy about. And this is, by any means, it's not a stagnant field. We have many different options and directions to go into. What I showed you today was more from a starting perspective and what we applied to. But we have here many different options for excitation, for inhibition, and for controlling of biochemical pathways as well. So this is what my lab does, builds the tools, and then we use them in brain circuits. This is my lab. I was fortunate to work with a wonderful construction team that created a custom-made lab so we can perform our in vivo experiments. And it allows us to go all the way from protein engineering to behavior control. And this is the brave team that joined the starting PI without knowing whether it's a good thing to do or not. So I'm very grateful for their trust. So our lab, if you want to visit, is Ali's second floor. And with this, I thank you, and I'll take questions.
What an information-packed talk. Thank you very much.
Well, I was sure that the audience can handle it, so.
Do we have any questions? Everybody hear that? Does any of this research give you insight into what's causing Alzheimer's disease and how to treat it?
We cannot get insight into what's causing it. we believe that this would be more of a biochemical approach. But we know that in the Alzheimer brain, because of the damage of the cells, there's perturbed activity in the circuits. And we have ways to intervene to restore that activity. So one beautiful paper from the Tonegawa lab used optogenetics in the hippocampus. And hippocampus is an area well researched for learning and memory, and it was shown that you can restore some of that activity. So while it doesn't give you information on what causes the disease or how to stop it, it could give you ways, just as DBS is a way to address the Parkinson symptoms, it can give you ways to address the symptoms. It would not cure the disease, but it could help the symptoms. Yes, you are correct.
Did everybody hear the question?
So
Some of the research appeared to be transgenic, and some of it involved transplanting viruses into the DNA of the cells.
yes. So when we use viruses, we use, when we started in the field, we were using lentiviruses because they could package more DNA. But now we moved away to adeno-associated viruses, which are more user-friendly viruses. And there's many clinical trials in advanced phases that use adeno-associated viruses to deliver other proteins to the human body. So it's a method that's being used in the patient population as well. And it's a method that we prefer in the lab. The drawback is that we cannot package too much DNA, so we are limited to short sequenced opsins.
You know, I think we're out of time. Dr. Gradinaro will make herself available in the lobby if any of you have any other questions. I'd like to thank Dr. Gradinaro for a very interesting lecture.
Thank you for listening.